Hello everyone, my name is Yuan, and I'm from Christina Leslie's lab in Memorial Sloan Kettering Cancer Center. So as we know, predicting drug responses to cancer cell lines has always been a topic of interest in the field of system biology. Today I'd like to introduce a joint learning approach over drugs that improves prediction of cancer drug response. By exploring large cancer cell line screening datasets where genomic and pharmacological data are available, it is possible to train models that predict drug responses to cancer cell lines. These models will not only help us understand more about the cancer genomics, but will also shed light on personalized cancer treatment and drug repurposing. Traditional approaches to drug response prediction are mostly single tasking, which means models for each drug is trained separately. Here we take a machine learning approach and ask the question, can joint learning with all drugs at once improve the performance of training, and can we learn models that make more biological sense? It is worth mentioning that winner of NCI Dream Drug Sensitivity Challenge in 2012 also used a joint learning kernel approach. We here take a different strategy that is straightforward, highly scalable, and quite efficient. So we first tested our approach on the Cancer Cell Line Encyclopedia, CCLE dataset. This dataset includes genomic and pharmacological information of 500 cell lines representing 36 tumor types. The genetic profile each contains 50,000 genomic features, including gene expression, mutation, and copy number alternation. Pharmacological profiling of 24 drugs is performed on these 500 cell lines in the form of activity area measurements. For the purpose of our learning, we have constructed a feature matrix and a response matrix. Feature matrix contains all the genomic information, and the response matrix contains all the drug response information. We want to learn a weight vector for each drug. So for the 24 drugs together, we want to learn a weight matrix that connects this feature matrix to the drug responses. And with this model, we can predict responses to new cell lines. In a single task scenario, weight vectors for each drug is learned separately through linear regression. Here, as a comparison, we have performed single task learning with ElasticNet regularization. So in ElasticNet setting, we want to learn the weight vector that minimizes the loss function plus L1 penalty and L2 penalty. L1 penalty is the penalty used in lasso regression. It basically makes the coefficients sparse, and L2 penalty is the penalty used in ridge regression. It pulls all the coefficients towards zero. By having this trade-off in ElasticNet between L1 and L2 penalties, we have make use of the usefulness of both, and this is the widely used method in system biology. On the other hand, for a joint learning, weight vectors for all drugs are learned at the same time, so we are learning this weight matrix. Thus, we try to minimize the loss function plus a regularizer on the weight matrix. Here, the regularizer we used is a nuclear norm regularizer. So nuclear norm is the same thing as a trace norm. It's basically the sum of all non-zero singular values. For a matrix, the number of singular values is the same as the rank of the matrix. Thus, intuitively, by putting a constraint on the nuclear norm, we are forcing the weight matrix to have a smaller rank. That is, the weight vectors of all these drugs have to live in a smaller space. And when they live in a smaller space, we are exploiting the connection, the relationship between drugs, so to help us increase the performance of prediction. However, joint learning is computationally very intensive. So to solve this problem, we have implemented alternating direction method of multiplier, AdMM. This is an efficient framework for breaking up optimization problems. The algorithm is pretty complicated, and I won't go into the details. So Yuan Paskov in the Leslie lab has developed an implementation for the AdMM as his Intel science project in high school. And his implementation in MATLAB is 10 to the 3 times faster than CVX, a general convex solver. And this has made our joint learning on large data sets possible and make them very scalable. So our joint learning is in a transductive cross-validation setting. And I want to make the distinction between an inductive setting and transductive setting for joint learning. As we can see here, here's the feature matrix and response matrix. As a reminder, each column in the response matrix represents one task. So in an inductive setting, the training set and testing set are split exactly the same way in the response matrix. As a result, the test instances can be held out in both the feature matrix and the response matrix during training. In the transductive setting, training and testing sets are split differently among different tasks. As a result, the test instances are still held out during training, but we can see the entire feature matrix. It is in the transductive case that we see this dramatic performance improvement, and this is the setting we used to report the following results. This is a figure for the cross-validated performance comparison between ElasticNet single-task learning and nuclear norm joint learning, reported in mean squared error. As we can see that there's a dramatic reduction in mean squared error for nuclear norm joint learning. The red bar is lower than the blue bar, and the average performance improvement is 35%. Which is quite impressive. And we have this performance improvement across all the 24 drugs. So as we introduced in the beginning, we think our joint learning model will not help us improve the performance of prediction, but will also make more biological sense. So here is a hierarchical clustering of drugs based on the weight vector, the W matrix learned from ElasticNet and trace norm. We can see with ElasticNet, the clustering is really flat. And so here, drugs belong to have the same mechanism of action, have the same color. So these drugs in the ElasticNet, drugs of the same mechanism of action does not cluster very well together. Whereas in the trace norm case, drugs sharing the same targets really cluster close to each other. For example, the Alatinib and Lapatinib are all EGFR inhibitors, and in the trace norm, so they are in orange. In the trace norm case, they are right next to each other, and in the ElasticNet case, they are pretty far away. So, of course, here in the CCLE dataset, we only have 24 drugs. However, since our method is very scalable, we can also apply it to a larger dataset, such as NCI 60. So for the NCI 60 dataset, I used transcription profile of 59 cell lines with 44,000 probes, covering 18,000 genes. And we also have pharmacological data for these cell lines for 169 drugs, either approved by FDA or currently under clinical trial. Again, we have constructed a feature matrix and response matrix. And feature matrix contains all the genomic information, response matrix contains all the pharmacological information. Again, this figure shows the cross-validated performance comparison between ElasticNet and nuclear norm in mean square error. So the blue line is the mean square error for the drugs learned from ElasticNet, single task learning, joining together. And the red dots are the mean square error for the drugs learned from nuclear norm. We can see that it's pretty obvious that most of the red dots lies below the blue line, which means that we have this performance advantage for the nuclear norm. For 133 out of the 169 drugs, joint learning performs better, and the average performance average performance improvement is about 16%. Here we made a heat map for the W matrix learned from ElasticNet. Each column is one drug and each row is one gene. Red means there's a positive coefficient, and blue means the coefficient is negative. Each drug is color-coded, and drugs sharing the same mechanism of action have the same color. Here we can see again the clustering is really flat, and drugs having the same target in general does not cluster together. Also, there's no clustering of genes at all because ElasticNet is, each drug is trained separately, and so they all live in different spaces. And here is a heat map for the W matrix learned from trace norm. Here we can see that the drugs have pretty clear clustering. Actually, we can split them into 4 different clusters, and within each cluster, drugs actually have mechanisms of action that are quite similar to each other. So here, drugs in the first cluster are basically mostly DNA damaging agents. Drugs in the second cluster are targeting proteins. The third cluster is kind of broad category, and the fourth cluster are mostly signaling agents. Similarly, for the genes, we can also split them into 9 clusters. And gene enrichment analysis is done on each of them, and gene ontology terms and their associated p-values are shown here. So on the next slide, I would like to go into the first drug cluster, the DNA damaging agent, to have a close look at this heat map. So in this cluster, there are mostly 4 categories of drugs. Drugs in red are topoisomerase 1 inhibitors. Drugs in purple are DNA synthesis inhibitors. Drugs in orange are topoisomerase 2 inhibitors, and drugs in blue are alkylating agents. As we can see that they are all DNA damaging agents. And the gene clusters that have very high coefficients for these drugs are the red cluster and the yellow cluster. So we did the gene enrichment analysis and look at the associated gene ontology terms. Some terms came up are mitotic cell cycle, DNA repair, and nucleotide excision repair. They actually makes a lot of sense because these are the terms that you would imagine to be associated with DNA damaging and repair. So in conclusion, our joint learning has improved the performance of drug response prediction. And this drug clustering based on our model also reveals shared mechanisms of action. In the future, we would like to apply our joint learning approach on the DREAM7 breast cancer dataset and probably compare the performance to the challenge participants. And in the end, I would like to thank all the people in Leslie Lab and also Chris Pascoe for their contributions to this project, and also the funding institutes. Thank you very much. And welcome any questions.
That's what I supposed to say.
Oh.
So we have time soon for some questions.
Hi, my name is Ahmad, and this is very interesting work and nice interpretability from the model. So I was just wondering, have you tried to compare this method with any of the multivariate regression approach that also exploit this multitask learning? Say, for example, this ElasticNet, you were giving the examples with single task learning. But as far as I know now, they have also developed this method which uses this multitask learning.
I think we have not tried to compare it to other methods of multitask learning, but I think that's a future direction we can try to.
Okay. And the other thing is that, you run this experiment in the transductive setting. So in transductive setting, which in machine learning we can say that it could be a semi-supervised learning. So is there any way to evaluate that the improvement in performance is not due to the more data available in the training, or it's due to the multitask learning? Is there any way? Have you tried that?
So actually, in an inductive case, the performance is quite similar to the elastic net setting. So in a complete inductive setting and in a complete transductive setting, there's this dramatic performance improvement. So I think this. I think multitasking has a kind of advantage, but this transductive setting has also gives us this performance advantage. And yeah.
Okay, so you are saying that this is because of the because of both of these things, the more training data available, and then you are doing this joint multitask learning.
Yes,
Okay.
I think so.
Okay, thank you.
Hi, my question is also related to transductive setting. In machine learning literature, transductive setting means seeing training data together with test data, but not seeing any of the labels. Actually, as a follow-up to Ahmad's question, I guess what you are doing is semi-supervised learning, not transductive setting. And the thing is that in order to compare with ElasticNet fairly, you need to include, for example, in your example, you have lapatinib and erlotinib, and these are EGFR inhibitors. And if you are comparing ElasticNet with your kind of approach, you need to include, for example, if you are training ElasticNet for lapatinib, you need to include erlotinib's response to the training data, right? Because your approach sees also erlotinib response, but ElasticNet didn't see that kind of information.
Right. I mean, we can. Yeah. I mean, for the ElasticNet setting, even we see the features that would not improve the performance, right?
My question is something like, if you have erlotinib response but not the lapatinib, right? You are predicting lapatinib response, but you expect them to have correlated values.
Yes, yes.
And if you see this value for the training test data, you can basically put the other drug's response to the missing value, and this will improve your prediction.
Yeah.
Right?
So I think as a validation, a future direction, maybe we can do like this kind of closest neighbor kind of training to compare it with our multitasking to see this improvement.
Okay.
Yeah.
A couple of questions. You went very quickly on this, but I assume you are training your system using the gene expression data and the mutational data pre-perturbation with the drug. So this is like
yes, this is all
et al. Paper,
yes,
etc. Right?
pre-perturbation.
So you're not using any of the data from, say, links or the connectivity map where the cells are observed after the perturbation.
For the NCS60, no.
So,
yeah.
so one question I have is that when people have used the pre-perturbation data, one phenomenal confounding factor is the lineage of the cells. And so essentially what you end up discovering as predictive element of why a particular drug works is that, I don't know, bortezomib works in, erythropoietic lineage cells, and, EGFR inhibitor work in epithelial cells, etc. So have you looked at that particular co-segregation, seeing whether you're corrected for that? Have you tried to see whether you can predict within a lineage the specificity of the drugs?
I think we haven't looked at a specific lineage yet, but using the breast cancer cell line data, maybe that will be a more specific tumor lineage we can look at, and that will be.
Yeah, I would recommend that you take a look at, so right now in the CCLE, there is a large representation, for instance, of ovarian, breast, lung cells.
Right.
So you can take those three contexts and see whether within each one of them you can actually stratify the response, because the problem is that I think you may have you may end up overestimating your predictivity simply because the drug stratifies so well with lineage. Okay, so that's something you may want to consider. And then the other thing that I didn't quite understand is your methodology right now. It's a little bit of a black box, at least I'm sure that when you look at the specific features that are used, you can actually identify specific mutations or gene expression values that are the most predictive. But if you open the black box to try and figure out exactly what are the genes that end up predicting specific activity of these drugs in specific cells that respond to the inhibitor?
I think that's what this heat map is trying to say. So this is the weight matrix that we eventually learned by using our model. And so...
No, I guess I'm asking something.
Yeah.
So this matrix here tells you what's the mechanism of action of, actually, what's the similarity between the mechanism of action of different drugs. But it doesn't tell you, among all the red bars, which one is most sensitive to a particular cell, right?
Not like in a cell-specific way, in a drug, more like a drug-specific way. So which,
right, this is drug-specific.
Yeah.
What I'm asking is, have you done this analysis to basically predict which drug will work best in a particular cell, right? And in that case, what are the genes that predict that?
Okay.
I thought that's what you were getting at, you know. So this is more like Mantra or some of the other algorithms that have been used to do broad analysis of mechanism of action or similarity of mechanism of action. But have you also looked at predicting sensitivity?
Predicting sensitivity for any of the cell lines. We haven't done that yet. Yeah.
Any other questions? Great.
So I declare the discussion started for the afternoon.